Hi everyone, welcome back to Pharmagist. Myself Dr. Haimavati. In today's video, we'll be learning about drug metabolism. So what is drug metabolism and what are the different aspects of drug metabolism? So drug metabolism is a very vast topic. So I have divided this topic into different parts, and this video will be the first part of drug metabolism, which contains the introduction and initial introductory concepts in the drug metabolism. Now let's begin our video. Now what is drug metabolism? Suppose you take any drug into the body, what happens? It gets metabolized by the action of different enzymes which are present in your body, and then it reaches the site of action. It performs its required action. For example, if you have a fever, you take a drug, so your fever will be reduced, and after the reduction of the fever, what will happen to the drug? It should get eliminated from the body. So it cannot stay in the body forever. If it will stay in the body forever, it becomes toxic to the body and it produces unnecessary or unwanted side effects. So that is why what we have to do, the drug, after performing its action, it should come out of the body. So this entire process, so the body, uh, how it performs, so in order to protect itself, so the body has its own mechanism to transform these drug molecules. So after their action is over, they are eliminated easily from the body. So this overall process is known as drug metabolism. So now coming to the definition of drug metabolism. Drug metabolism involves the biotransformation of drugs or non-essential exogenous compounds in the body so that they can be easily eliminated. So this is the actual definition of the drug metabolism. So I have explained just now, no? So what happens whenever you're taking the drug, it should be eliminated. So how the drug gets eliminated from the body, so that process is known as drug metabolism. It is also called as xenobiotic metabolism. So by the process of xenobiotic metabolism, what happens? The drugs are made more polar. So this polarity is brought about by introduction of polar moieties into the drug molecule so that they can be easily eliminated from the body. So this drug metabolism or xenobiotic metabolism, what it does, it makes the drug molecule more polar by the addition of a polar moiety such as a hydroxyl group or an amino group, so that it can be water soluble and it will be easily eliminated from the body. And liver is the major organ for metabolism. Now coming to xenobiotic metabolism. So what is xenobiotic metabolism? So our human body is exposed every day to a number of foreign substances like the dust that is around us, the food we take, and the pollutants which are present in the air. So all these are nothing but xenobiotics. So anything which is not a part of the body is called a xenobiotic. So all these foreign particles are collectively called as xenobiotics. So the body cannot synthesize its own food or cannot synthesize all these things. So anything which is not from the body is called a foreign substance. So foreign substance is nothing but a xenobiotic. So these xenobiotics, when they enter into the body, so our body has a protective mechanism so that it modifies these particles and so that they are excreted from the body. So this process is called metabolism, and overall it is called xenobiotic metabolism. So drugs are also foreign substances. So because they are foreign substances, they also come under the category of xenobiotics. So that is why drug metabolism is also called as xenobiotic metabolism. And it is also called as biotransformation. Biotransformation means the drug molecule also gets transformed. So the different functional groups change into another functional group. So transformation of the molecule also takes place by the action of the different enzymes which are present in the body. So drug metabolism can also be called as xenobiotic metabolism and also biotransformation. And so this process of metabolism, what it does to the molecule. So anything, three types of outcomes can happen for these drug molecules. So first one is the drug molecule then can become totally inactive. So here the first one is the drug molecule is made totally inactive. So it won't have any further action. And second one is it can produce toxic metabolites, and third one, it can produce active metabolites. So active metabolites means it is a prodrug concept. So the drug which you are taking into the body, after the action of these enzymes, what happens, either it will become totally inactive, or it may produce any toxic metabolites, or it can produce any active metabolites. So the drugs which undergo metabolism to produce a pharmacological effect are called prodrugs. So if the transformation or the drug metabolism brings about an active molecule, so this active molecule will have a pharmacological effect. So that is known as prodrug. For example, phenacetin, it is metabolized into paracetamol, acetaminophen. So phenacetin is a painkiller, paracetamol is an NSAID which is used as a anti-inflammatory agent as well as antipyretic. So paracetamol is more safer than phenacetin. So initially phenacetin was used as a painkiller, but now it is banned because of its toxic side effects. Whereas paracetamol, we are using it as an NSAID. So phenacetin is the prodrug for paracetamol, and levodopa is the prodrug for dopamine. So levodopa gets metabolized to dopamine. So why this concept of prodrug came? Because of first pass effect. So what is first pass effect? So some drugs, when you take them into the body, what happens? Before they reach their site of action, they are metabolized. So as a result, what happens? They cannot show their action on the required organ. So then what happens? The drug will not be able to show its effect. So we are at a disadvantage. So for that, what is done? So the concept of prodrug was evolved. So in prodrugs, what happens? The drugs which are prone to this metabolism, they are masked. That means we add some additional functional groups, so to prevent the hydrolysis or metabolism by the different enzymes before they reach their site of action. So as a result, what happens? That drug, after it is metabolized, it gets into the active form, and then it can show its action at the desired site. So that is how the first pass effect helped in designing prodrugs. And another thing is the drugs, they can undergo metabolism to produce toxic metabolites. So toxic metabolites, they can be either carcinogenic or mutagenic. So there are three outcomes of a drug metabolism concepts. So either the compound will be inactive, or it will be carcinogenic, or it will have a pharmacological effect. And drug metabolism also occurs in the kidney, skin and gastrointestinal tract. So apart from the liver, metabolism can also take place in kidney, skin and the GI tract. Now we have different biochemical reactions in drug metabolism. So the overall drug metabolism takes place by the process of different biochemical reactions. So these biochemical reactions are, first one is microsomal oxidation, second one is non-microsomal oxidation, third one is reduction, fourth one is hydrolysis, and fifth one is conjugation. So the overall drug metabolism can take place by any of these five types of biochemical reactions. In the liver, we have something known as the microsomal oxidation. This microsomal oxidation takes place in the mostly in the liver. So in this liver, we have cytochrome P450 family of enzymes. So in the cell, there is a smooth endoplasmic reticulum. So when you study about the different parts of the cell, you have studied something about smooth endoplasmic reticulum. So this smooth endoplasmic reticulum contains a family of enzymes which are known as cytochrome P450. So these cytochrome P450 enzymes, they bring about the oxidation. So that is why it is known as microsomal oxidation. So this cytochrome P450, they catalyze the oxidation of majority of the drugs. So most of the drugs that we take into our body, they are metabolized by this cytochrome P450. So in this cytochrome P450, there is a porphyrin ring which contains a ferric ion. So ferric ion means Fe3 plus. So this Fe3 plus is reduced to Fe2 plus, reduced or changed to this 3 plus, 2 plus. Okay. So in this cytochrome enzyme, what happens during the this metabolism process? Transfer of electrons takes place due to the enzyme known as NADPH cytochrome C reductase. So this NADPH cytochrome C reductase, it supplies the electrons for this cytochrome enzyme cytochrome P450, and overall process is an addition of oxygen and electrons takes place, so that there is formation of a hydroxyl moiety and water. So S, here S means it is the substrate, that is the drug molecule. So in the overall reaction of this microsomal oxidation, what happens is, to the drug molecule, one molecule of oxygen is added and two electrons are added. Ultimately what happens is, an OH group is added to the substrate molecule and a water molecule is formed. So this is the cycle which you can see here. The substrate, it when it attaches to the cytochrome P450, this Fe3 plus, by the addition of electrons, it gets into Fe2 plus, and then an oxygen molecule is added, and finally what happens here, you are getting a water molecule and you are getting the oxidized substrate, that is a OH molecule is added to the drug molecule. So now this is the structure of the cytochrome P450. So this I have downloaded to show you. So this is just for information purpose only. So this is not a part of syllabus. So scientists who have crystallized the cytochrome P450 enzyme, so they have deposited in the protein data bank. So in this protein data bank, this is the cytochrome P450 enzyme structure. So this is the three-dimensional structure of the cytochrome P450 along with the porphyrin ring. So here this enzyme has a porphyrin ring in this molecule. So you can see this. So the porphyrin ring, this is the 3D image, and on the left is the 2D image. So this one is 2D image. So when you visualize it in the software, it appears like this. So this is the crystal structure of that porphyrin ring. So here you can see in the center of the molecule there is a Fe3 plus. So during the drug metabolism, what happens? This porphyrin ring, it attaches to the drug molecule, and this Fe3 plus becomes Fe2 plus, and the drug molecule, what happens? There is an addition of an OH molecule and a water molecule is also formed in the overall reaction. And next we have non-microsomal oxidation. So what happens in non-microsomal oxidation is, there are some more soluble enzymes which are present in the cytosol or the mitochondria of cells. So these enzymes, they are responsible for the metabolism of some of the compounds. For example, we have alcohol dehydrogenase and aldehyde dehydrogenase. So this alcohol and aldehyde dehydrogenases, they oxidize ethanol to acetaldehyde and acetate. Okay, so these dehydrogenases, what they do, they specifically oxidize the ethanol into acetaldehyde and acetate. Then we have xanthine oxidase. This xanthine oxidase, it converts hypoxanthine to xanthine and then that xanthine is converted into uric acid. So tyrosine hydroxylase. So this tyrosine hydroxylase, it converts hypoxanthine to xanthine hydroxylase, and then hydroxylates tyrosine to dopa. And then we have monoamine oxidase. This monoamine oxidase, it is responsible for the metabolism of catecholamines and serotonin. So this is about non-microsomal oxidation. So some certain set of enzymes are there. They specifically metabolize these compounds only. And then we have reduction. So reduction, it occurs in both microsomal and non-microsomal metabolic systems. So under the microsomal reduction, what happens is reduction of nitro groups and azo compounds takes place. For example, chloramphenicol is a molecule which is having a nitro group, and then prontosil has an azo group. So these nitro and azo groups, they are reduced into more polar groups. And then we have non-microsomal reduction. So the examples of non-microsomal reduction are reduction of aldehydes, ketones and quinones. For example, chloral hydrate is an aldehyde, and naloxone contains ketone groups, and menadione contains quinone. So these molecules, they are metabolized by the process of reduction reaction. Then we have hydrolysis. So this hydrolysis, there is non-microsomal hydrolysis which are present, and they are also present in the plasma also. So they are non-specific sometimes. For example, acetylcholine is a non-specific esterase, and then we have peptidases, for example proinsulin, and then we have amidases. Okay, so these non-specific enzymes, and they bring about non-microsomal hydrolysis of these particular molecules. And then we have conjugation. So conjugation means coupling of a drug or its metabolite with an endogenous substrate. For example, a carbohydrate, a sulfate or a methyl group to make the drug more polar and readily excretable is called conjugation. So we have different types of conjugation. For example, glucuronide conjugation, sulfate conjugation and glycine conjugation. So in conjugation, what happens is coupling of the drug with an endogenous substrate. So a substrate which is produced by the body, for example a glucuronide. So these glucuronides, they are produced by the liver. So they get attached to the drug molecule. Or example, a carbohydrate molecule or a sulfate or a methyl group. So what happens is these molecules, they bind to the drug and make it more polar and more water soluble. So this is known as conjugation. So coupling of a drug with its endogenous substrate is called conjugation. So these are the different biochemical reactions which are involved in drug metabolism. I hope the video was informative to you and useful. So in the next video we will see some of the examples of drug metabolism as well as phase 1 and phase 2 reactions.